Good afternoon, you all. Thank you for coming on a Friday afternoon. We have a very special treat for you today. Dr. Fabiany Herrera from the Chicago Field Museum came all the way down to visit us and talk about tropical rainforest. So give it a little bit of background about him. He trained as a geologist starting in Colombia, went up to University of Florida, trained a little bit harder, then switched over to botany. And so through that amalgamation of training, became a paleobiologist and has been discovering fossils all over the world from Mongolia to Peru, and of course the Great Plains of Illinois. And without further ado, I'll let Dr. Herrera take it off.
Thank you so much for having me. Go Gators. Oh, wrong school, sorry. Just kidding. What do you guys say here?
Panthers. We're the Panthers.
Wow. All right. No, I'm really excited to be back in Florida, and today I'm gonna talk about fossil plants. And this is a 20-year history for me since I was an undergraduate student, just like many of you here. I was just a curious student, a geology student, and I love fossils, and I wanted to study all of them. Ultimately, I chose plants, or they chose me, and that's the work that I'm going to show you today, my last 20 years. But what is really important is the 300-million-year-old history of the tropical rainforest. I'm originally from Colombia. That means I grew up surrounded by mountains and forests. And I was fascinated by that. I think that was one of the reasons that I decided to study geology in the first place. It was when you go to my hometown, we had a mountain range that it just covers everything. And that's what
What is your hometown?
Bucaramanga.
Bucaramanga.
Which is in the northeast part of the country. And there is an incredible forest on that mountain that goes from near lowlands all the way to near Paramos in that part of the country. Absolutely beautiful. And understanding the evolution of two is what I'm ultimately fascinated by. Now let me tell you a little bit about plants, because as I was introduced, I'm a geologist, but I'm also a botanist. And today, we only have, we had about 370,000 seed plants. That means plants that have a seed. So they are basically not ferns that they reproduce by spores. And these seed plants can be grouped into 5 groups. Imagine that, just only 5, which is a relatively a small number of seed groups. And what is more interesting about these, which includes, for example, Taxaceae, gymnosperms, or conifers, what we call cycads, that are very common here in Florida. We have Nitalis, this weird group of plants, ginkgos, the only surviving group plant of a very long, a group with a long evolutionary history. And angiosperms, these are actually photographs from Colombia. If you get a chance to go there, please go. And you're going to see that the flower marker is just an incredible amount of species. Many of those flowers, ultimately, you can buy at Publix. And the fruit market, of course. But understanding the deep relationships between these 5 groups is absolutely fundamental, because we need it tells us the history of plant evolution. But what is important for me is we need fossils to fill the gaps that ultimately can help us understand how they relate to each other. This has become an iconic figure in paleobotany, and it's very simple to look at. What I want you to see is time on the top. Here from the Triassic, we see 2 mass extinctions here. The last big one, the late Cretaceous extinction. And here what we have is biodiversity. And these 2 peaks that we see here is this battle between conifers and angiosperms that has occurred over the last 150 million years. And what we see is that during the Cretaceous, there was 2 peaks in diversity. These 2 groups had an increased diversity here. And this has been also represented here in sort of a gymnosperm dominated ecosystems to ultimately this battle was won by angiosperms that dominate the rainforest today. Now, the other important part about plants is time, of course. And here I have a few paleogeographic maps that show 300 million years up to 50 million years. What I want you to pay attention to are the stars, these red stars here. It's just showing up what would be the tropics today in this part of the world, basically Colombia, Panama, Ecuador, Peru, all that region. About 300 million years ago, Chicago occupied this land, this part of the world. And then here we have now the northern part of South America. It literally has stayed in that region since the last almost 200 million years. Africa separated, Central America did not exist, and then ultimately it stays there for a long period of time, which made these plants very interesting because they really tell us about how the tropical rainforest from this part of the world has evolved during the last 200 million years or so. And the question that I want you to keep during my presentation is, have the tropics always been a cradle of plant evolution? No matter what part of the time, and plants can help us to understand that, fossil plants. All right, let's go to Paleozoic. As I mentioned, Chicago was right here, and it was not only at the equator, it was also very close to the ocean. And for me, every time I think about Chicago in the tropics is when in the middle of the winter in January, and I'm like suffering and freezing my butt with this horrifying winters. And the fossils that left, that were produced in this time come from a place called Mazon Creek, about 310 million years old. It was a tropical shore. They are found in this Mazon River. It's just 1 hour south of Chicago, and historically has been a very important place for Illinois and the Field Museum because most of our collections are represented from this deposit. This was a tropical shallow marine system where we have an incredible amount of fossils, mostly vertebrates, includes very primitive sharks, fish, other snakes, early tetrapods, a coastal forest, and this guy here that is also an icon for Illinois is Illinois state fossils called the Tully monster. Every year there is a different paper. It is a vertebrate, it's not a vertebrate, at this point I don't know what it is. But it's a really cool fossil. The eyes are here, the mouth is here. Anyway, but plants are very important. We have more than 20,000 catalog specimens. That means that we still have lots of fossils that we need to catalog and put in our collections. And every year we have amateur collectors that donate fossils to our collections. And when I started my job at the Field Museum about 3 years ago, I wanted to study this material because no one has done it for decades. and I'm also a paleobotanist that I like to use all kinds of modern techniques to restudy this material. And many of them have remained poorly understood. Also, one of the things that when you notice these fossils that are formed in concretions, here they always have these egg-shaped structures, is that you can see some three-dimensional in preserving them. And in terms of the composition, we have lots of plants. Most of them has gone extinct. A few of them still have some remaining survivors. And the old-fashioned way to study these Mazon Creek fossils was by cutting the specimens. I mean, using a saw, you cut it in half in multiple pieces, you photograph it, and then ultimately you're trying to do a 3D reconstruction based on that. And that is a destructive way to study the fossils. And I'm not blaming my previous colleagues for doing that. It was just the only way, and that's still used today, the only way to study them. Now, where to begin? 20,000 specimens. Imagine the size of the collection. Which one should I pick? What should I prioritize? What is gonna scan well or not? And that's what I wanted to do, is CT scan this material. So I chose two of the enigmatic specimens, something called Tetraphylostrobus and Crossoptica. And for the last 10 years, we've been using the CT scanning at the University of Chicago. Now we got 2 scanners at the Field Museums. If you're gonna study new material from there in the near future, we are gonna be able to scan fossils and living specimens. I'm not gonna go into much details, but it really takes a lot of energy and time to understand what is good. And voila, the scan came out better than I ever expected. So this is the Tetraphylostrobus right here showing the internal anatomy of the specimen. Here, what is really nice is that it allows you to see the face that is buried within the rock, and you can make a reconstruction. Basically, it's like having Superman vision, X-ray vision, and you can flip the specimen. And I'll talk about this in more detail in a second. This is the other specimen that look very, it's hard to see, to be honest, with the naked eye. It's like, I know there is a plant there, but then it's when you see the digital section, how clear it becomes. And then the reconstructions, which is really fascinating. Now we can see these plants in 3 dimensions, an incredible detail. This is a spore organ, so all the spores were located inside these structures. Now the question is, 20,000 specimens, what do I do? What's still good? So I looked at the mineralogy, the composition, and by this is I use in situ Raman spectroscopy, which allows me to see what are all the minerals within the fossil. And then that I compare it with the results that I was getting with the CT scanning. And this is some of the results. It tells you basically, this is also some EDS maps with the SEM. So I combine both analyses, and definitely the mode of preservation is going to impact the way I look at the fossils. And this is going to be fundamental for the type of studies. Heavy minerals are going to provide the best results. Silicates or clay minerals are the worst. Right here can have mixed results. And this is more of a methodological paper that I wanted to show. But if you want to learn more about how to CT scan, that's the paper that we published last year. And now let me tell you about Tetraphylostrobus, and this is a work that I've been doing with my postdoc, Mike D'Antonio. So one of the things is that when it was first published from Canada, this genus, the guy who described it, what he saw is that there were 4 scales, 1, 2, 3, and 1 in the back that you don't see. And he created this model. And this is not the type of plant organization that we see in the Paleozoic. In fact, never seen it. This is what plants do today, but not in the Paleozoic. They all form sort of worlds and things like that. And that's what this fossil has been placed in every single group of plants, and no one knew what it was until this year. And that's because instead of 4, it has 6. And it makes total sense. And here you see 1, 2, 3, 4, 5, 6 in the back. And we see it also in transverse sections. So we solved the mystery, 40 years in the making. So we were finally able to resolve it as a group of Xenophiliales, and I'm going to show you what they are. And we not only were able to see the structure, we saw all the spores, every single detail of this plant. But imagine again, this is a 300-million-year-old plant. This is a reconstruction, by the way. Excuse me for the selfish promotion. My niece made this reconstruction. If you ever need a notice, let me know. But this is what Xenophiliales look like. This is one of those typical flower-like leaves that we see very common, Sphenophyllum leaves. And now we resolve the mysteries from this plant 300 million years ago. But this is just the beginning. What I'm really excited about is all the new opportunities that CT scan is going to allow us. This is, for example, a primitive type of seed, Rhabdocarpus. That's the CT scan. We can see every single cellular detail. And again, when I see Chicago, especially the skyline here, compared to those giant lepidodendron trees, I wish we will still have them today, or we could travel back in time. But I highly recommend that you come and visit us and see this collection at the Field Museum. All right, now I'm going to take you to the Mesozoic, which in this time, Colombia and Ecuador, they were sometimes covered by the shallow epicontinental ocean. And, but basically, we were right at the equator at this time. And one of the things that we learned always in school is that most of South America was a very dry place, which doesn't make sense, right? Or at least it's not something that we expect. And this is also a sort of typical paleoclimate, paleovegetation reconstruction for that period of time during the Early Cretaceous. Tropical Africa was very humid, parts of Europe. It was warm and temperate here in North America, but very dry in this place. And the reason I can tell you this is because we have fossils, lots of fossils that come from Crato, Brazil, that have dinosaurs, pterosaurs, and it was based on the composition of the planet. That part of Gondwana was likely very dry, but we don't really know about the rest. So we made lots of assumptions based on a single locality. So during the last 10 years, I've been visiting with my students and other colleagues, lots of localities. Many of them are historical localities known for decades, but poorly studied. And others are completely new to science. So we have localities that go basically from the Palanginian all the way to the Cenomanian. And the first one is what we call the Paja flora. This is from the central Colombia. This area is known for marine vertebrates. We have from this big marine reptiles, some of the earliest marine turtles, lots of ammonites. Funny fact, these fossils were mostly studied by priests. He was the only paleobotanist we had in Colombia in the 70s, all the way to, I became the second one, I guess. So it's kind of funny. He was great collecting fossils, but not so good identifying them, unfortunately. But I had a lot of respect for him anyway. And when you see this reconstruction, is that in the back we have plants, but to be honest, people just use, let's just put araucaria trees in the back because they were likely there, but we don't really know. So with my student Hector Palma, he started his PhD at the University of Illinois in Chicago this fall. We've been revisiting lots of these small museums all over Colombia to collect fossils and study them. We have so many fossils from this locality that fences in these villages are completely covered by ammonites and fossil plants. And I've been tempted to get a hammer and go at night and get them out, but I don't want to be killed. But the fossils are incredible. Look at this. This is just a view of a couple dozen specimens. They're three-dimensionally preserved, and they were preserved in a shallow marine deposit, which it has some problems to reconstruct the forest, but it's giving us an idea of what the forest looked like during that time. We have very interesting plants. I'm going to show you more details, but some of these cones are about the size of my head, almost like a pineapple, which is really amazing. We never really studied them. But let me give you a short story about these fossils described by the priest. He described this one as a sphenophyllum. That's a sphenopsid that I just showed you from Mazon Creek. Somehow he saw the fossil. These are Mazon Creek fossils, by the way, for comparison. And this is what he reported in his book before he died several years ago. And he described this as the Sphenophyllum colombianum, which makes no sense because the group likely went extinct during the Permian. But how do we have this here? And that's when I finally got a hold of the specimen right here. I see the resemblance, but I knew immediately this is not a plant. And I had no idea what it was. And this is, if you're gonna be a paleontologist, you need friends. And when you don't know what it is, ask your friends. And I sent it to my colleague in Colombia, and he knew immediately, it's a baby turtle. So instead of it, it's a Pokemon actually. And my students, of course, are huge fan of the Pokemon, and they name it Turtwig because apparently there is a baby Pokemon that is half a turtle and half a leaf. So we give it a nickname. And but more importantly, funny fact is we really need to restudy this material because we made mistakes. So it's a baby turtle, but now what about the plants? So this is one of the ones that my student is studying. Now this is an arborescent fern here in cross section. This is the specimen is quite big. Arborescent ferns were pretty common during the early Cretaceous. And here we might have a new type of genus, might look like something like this that has been reconstructed from other parts of the world, which is telling us a little bit about the environment. And I'm just going to show you now briefly some of the beautiful specimens we had in this material. Loss of foliage. It CT scans beautifully, so now we can look at it in 3 dimensions. Priest Huertas called this thing a passion fruit. which is stuck in the literature for decades. It's not a passion fruit. It's actually a group of gymnosperm that live in India and Australia at the same time. Now we have it for the first time in South America, expanding the range of this group. And the Araucariaceae, that family, that conifer that still lives in Chile and Argentina, was the dominant group at this time, which is amazing. Think about this. Today, it's dominated by gymnosperms. So this is a type of forest that we have no analog today. Nothing like that. All right, now briefly, I'm going to show you the other two localities, which I'm very excited about. One is from Ecuador. This is Ojin, and the one Une is from Colombia. And both of them, they don't have the same type of preservation, but like the Ojin in Ecuador, we have amber, which is probably the largest amber deposit in South America. And that amber was most likely produced by the Araucariaceae trees or the monkey fossils. And we're finding even insects, just like the Jurassic Park movie. Ferns are very common. And most importantly, all the 5 groups of seed plants that I told you about it at the beginning, they are all coexistent at the same time in the same type of forest, which today we have no analog for it today again. So lots of different plants from this time. And more importantly, the earliest angiosperms from this part of the world. We never seen them in macrofossils. Now we have them here as leaves. So this is going to open up lots of opportunities to understand what kind of forests, what kind of, when angiosperms appears at this period of time. Remember that peak of the figure that I showed you? These fossils are going to help us hopefully to resolve this. We've done a lot of palynological work from the same localities, which indicates that angiosperms were definitely present and all the other types of seed plants. This is the other Cenomanian uniflora with the same 5 groups, Araucariaceae again, angiosperms. But now, for the first time, the first flowers. We've never seen flowers from this part of the world. And these are like little lamps that are shown here with fluorescence. If you look at this with transmitted light, looks like nothing, just some piece of a plant. But when you turn on the fluorescence, all the anthers here and all the pollen, it shows up in beautiful details. So we're gonna, still a lot of work to do. But here, just again, that question that I asked, has the tropics always been a place of origination in terms of plants? Maybe it is. But more importantly, in the future, I hope to revisit this hypothesis of the arid concept for the entire Western Gondwana might be more complex than we think. And all these ferns that I show you might indicate that it was likely wet. What about CO2? What about the type of forest? Those are questions that I hope to answer in the future. But more importantly, this is going to give us a first picture of what the forest might have looked like during the early Cretaceous. Well, I'm going to do a break here. I'm going to take you to Mongolia. Well, I'm going to take you to Mongolia because I've been there collecting fossils, but also because these are about the same age of the ones that I just showed you. So I want to illustrate what was happening on the other side of the world. And if you go to Mongolia, which here is Russia, here is China, here is the Gobi Desert, this Google map just tells you no plants today. It really is very dry, high mountain desert. But and in the middle of the country, we had this little tiny coal mine called Gobi, and we visited it several times. It is a student for scale bar, and this is a lignite, which means it's a low type of coal or a very low grade. In other words, it's very dirty when you burn it, and that's the only way. That's the only energy we can get from this part of Mongolia. But the plants that made them are exquisitely preserved. These are the fossils, but we don't collect them like that at first. We collect the lignite, which looks like this, and then we put it in soap and hot soapy water, and that makes the lignite to go in an epileptic attack, I guess, something like that, and all the fossils separate when we let it dry for several weeks, and then we get this, and that's where the fun begins, and we start to collect all the things that we have found. And this is a filmy fern. Here is the fossil, the living one, the earliest of this genus, never seen before. Club mosses, petrify in this case, and other weird plants like this ancient relative of ginkgo that somehow survived in Mongolia. I wanted to show these figures because now we're looking at all the type of fossils that we get from plants, which is fungi. And what we have here, all these dots, is what the fungi attack the plants. And again, with fluorescence, we can turn on the light and you see all the hyphae and all the plant fungal interactions that happen during the Cretaceous here. So imagine, have you seen that TV show, the cordyceps attacking humans? That happens to plants. Not at the same scale, but that's what we've been seeing here. And all the fungi preserve the spores germinating and attacking all the plants, which is I talked to some students that are looking at all the fungal diseases in avocados and mangoes today as well. So here we have a forest with no angiosperms during the origin of the angiosperms, which is likely the last forest that prevented angiosperms to be part of the ecosystem. And we're still working on a reconstruction. All right, let's go back to the neotropics again. And for this now, we're going to the Cenozoic. So we have moved a lot of in terms of time, but here, and again, this is when I go, what make me a geologist actually is looking at all the mountains and all this complex geology that we had in the northern part of North America, South America. And when we think about the early Cenozoic particularly, we have to recognize a few things. There were no Andes. This is a completely lowland area. The Amazon River did not exist, or at least it did not have this, the current pattern west-east. And South America looked like a giant island. It was like, imagine Australia today, an isolated landmass compared to the rest of the world. And one of the places, this is actually the first place that I visited as an undergraduate student collecting fossils. And it's a giant coal mine in the northern part of Colombia called Cerro. And just to give you an idea of the scale, but this is the field museum. I could lay out 100 buildings at the bottom or more. Just, it's a giant hole that we created. And I know we have issues with in terms of conservation and stuff, but as a paleobotanist, this is a gold mine. And I've been lucky to be there, being one of the first paleontologists to look at these fossils. And other places near Bogota, and this formation is very different. These are mostly paleosols, and all this clay is used to build entire cities. In fact, if you go to Bogota, the entire city is made out of bricks that come from Paleocene rocks. And so it's amazing, the whole city. And the colors that they made, all these oranges and yellows, it's really beautiful. But when we started collecting plants, by accident, we also found some large, weird animals within the coal seams. And at first, we had no idea what this thing was. And here, until, of course, once again, we asked someone that knew, and he identified it. Oh, that's a snake, but not just a snake. The biggest snake that ever lived came from this locality. And this is what we call Titanoboa back in 2009 when we reported it. Why a snake got so big in the early Paleocene? Because the planet was super hot. Snakes are cold-blooded animals, and they need temperature to regulate their body temperature. And the only way to explain a snake of that size, here is a 12 feet living anaconda for comparison with the vertebra of Titanoboa. And the only way is to explain a super hot world. But what is interesting when we think about global warming today is that the forests survive, and not only survive, it flourish in a way. So here we have a rainforest, a tropical rainforest with giant animals. And more importantly, it's not the earliest rainforest, but it's also the earliest that look like the ones that you will see today. The monkey fossils were gone, monkey fossil trees were gone, all these ginkgos, tropical ginkgos, extinct, everything is gone. And here we start seeing the things that you recognize today, like the avocado, like the chocolate families, the legumes, the green bean family, the mesomorphic. Here we have palms, gingers, ferns, all those plants that we see today appear here. Another thing that we noticed is that there were very insect herbivores, and we know that because all the holes that you see here, in many of those cases were done by insects. So low plant diversity. Everything looks like a rainforest today from that part of the world, but not too diverse in terms of plants and insects. So that was a big question of we can understand it. So we might probably monitor Carvalho. All right, let's get more fossils. We need to answer that. So we visited more clay pits in Colombia, and not only of that age, but also before the extinction of the dinosaurs. And that's when we realized that the reason we have a new type of forest at that time and low plant diversity is because of most likely the extinction of the dinosaurs. So we collected fossils before and after the extinction. And when we compare Cretaceous versus Paleocene floras, here in green we have late Cretaceous, and in orange we have Paleocene size. These are all the main group of families or angiosperms that are present today in tropical rainforests. So the queen of the jungles are green beans, basically. That's the most important tropical family, followed by palms, coffee, moraceae, and other avocados. And when you see these records in the Cretaceous, very little plants. To be honest, if you show me one, I just can tell you, that looks like an angiosperm. That's it. But when you go to the Paleocene, oh, that's most likely that family. And that has a profound implication for understanding the evolution of rainforests today. Then we estimated, and we use mostly pollen in this case, that 40% of the plants completely die out during the extinction. And insects were also critically affected. Before the extinction, all the herbivores were specialized. That means that they only fed on a specific types of leaf. After the extinctions, anything, I'll eat anything. So they went from picky to not so picky after the extinction. And the forest, the structure of the forest completely changed. We realized that before the extinction, the forest was open or openish in a way. And we think that it was likely dinosaurs that were roaming through the forest and knocking down trees, like elephants do today in Africa and Asia. But if you eliminate all of those, the forest closed. And not only closed, but it developed many layers, low layers, and other plants that now are climbing to reach and compete for light. If you want to learn more about the extinction, just go to YouTube and Google what happened in the tropics during the last great extinction. We made a cool animation for this research. But let me show you the fossil, the king of the jungle. It's the most important Paleocene fossil in Northern South America. You see this is literally like a green bean that we're gonna be eating in a few weeks in Thanksgiving. The seeds here is beautifully preserved. The passion fruit, this is the earliest passion fruit. Just thinking for right now, a mojito passion fruit. But we had the oldest fossils here for this time. The custard apple family as well. And grapes, which are a very important group today, but particularly economic grapes. This is the first time that we see grapes in this part of the world. In fact, we just discovered the oldest grape from the Western Hemisphere. Now, two groups I show you here, grapes and passion fruits. If you like gardening, you know that these plants are epiphytes or are lianas, or they grow on top of other plants to reach the light. We don't see these fossils before the extinction. That means that the structure of the forest really changed. These plants only appeared and became dominant after the extinction. Another cool thing of this story of the grapes is that we show not only 60 million year old grapes, but all the way to the Miocene. And what we show is that this group, even though appeared in at this time, it has lots of local extinctions in the neotropics, which is completely unexpected. We always think that because it originated in a rainforest, it's gonna stay there forever. And that is not true. That's what the fossils are telling us is, wait a minute, something happened here during a particular time that I don't wanna live here anymore, and it went extinct. Why? I had no idea. But we're just starting to see this pattern. This study was also a battle. It took me, I literally fought a battle with the journal and fighting with the reviewers to get it published in Nature Plants, because I got a lot of criticism from molecular biologists because I made a critique of that work. When you use molecular data, you create a phylogenetic tree like this here. And then you can use different models to predict where the group originated. If you only use molecular data like this color shown here, here it shows you this is where the likelihood of where that group originated. In this case, I'm using this group with these fossils that I have here. And if I use all molecular data, all of them are telling me they originated anywhere but the neotropics. But guess what? The oldest one come from the neotropics. And that happened with every single fossil. That means that we truly need fossils to understand the origin of these groups. And cheers. Now, I'm gonna make another gap here and I'm gonna take you to Wyoming. Now it's slightly younger, about 52 million years old. And this is a place where I've been starting collecting fossils from the Green River Formation. And these places are absolutely amazing because all the work that has been done by amateur collectors, by private collectors, and institutions like the Field Museum. We take high school students every summer for the last 20 years or so. And now I'm in charge of this class, and we're collecting lots of fascinating things, mostly fish. That's one of the groups. And I have to acknowledge here Lance Grandi, which retired a couple of years ago. He's still active at the museum. He studied fossil fish from Green River, and he has a wonderful book. If you want to learn more about this, I highly recommend it. But Wyoming was located right here. And not only was very warm, but there were large lakes in this part of the country. And one of these, what is called Fossil Lake, an incredible fauna and flora. Fish, like I said, is the most typical type of fossil. We can find it in all types of preservation, completely exploded, completely articulated. The preservation is just incredible. This is one of the most important places of this age in the world. Birds with feathers, even with fruits. Now we're seeing inside their stomach. The earliest bats and tiny horses. It really is amazing what we see. But plants is my thing, and we can get some complete palm fronds. That's Lance Grandi, by the way. To a paper that we just published last month. And this is, once again, showing you why we need fossil plants. Yams. This is a very important plant in the world. It's one of the most important crops, actually. Entire nations in Africa depend on yams. It's not the sweet potato, although it might look like the same. And again, based on molecular data, it's been predicted that originated in Asia or Africa. We got the oldest, earliest fossil in Wyoming, of all places in Wyoming, once contradicting our knowledge. So this was an incredible place, a lake with tiny horses, the earliest bats, birds, a really fascinating place. All right, now let's go back to the neotropics. And in this case, now we're gonna go to Panama, about 40 million years ago. And here, Central America was not fully formed. It was just a bunch of large islands. It's still in the proto of what is the Pacific, or something in between. And it was at this time that volcanoes rose above water. And the first land appeared in this part of the Central America. And no, surprisingly, plants show up. And this is one of the best places to do field work, because at the end of the day, we have a high tide, so we can go in the collect more rocks. It's literally by the ocean. So you go and get fish and chips and a cold beer. But here on the beach, all these are fossil plants. All this is fossil wood, seeds, fruits. And they were early described by in the 1930s, but no one visited it. So this is one of these historical places, and lots of fruits and seeds. Some of them occurred in the neotropics. And again, lots of things that are only in Africa and Asia. Why they went extinct, I still don't know. In Peru, I visited this locality. Now this is a slightly younger, right on the northern part of Peru. Here are the Andes, so this is on the foothills. And with my PhD advisor, when I was a student at the University of Florida, it was first described in 1927, and it got lost in the literature, and we had no idea how to find it. We literally went to look for the needle in a haystack. And it took us about 3 days, and we relocated the place, and the fossils are just amazing. And again, passion fruits, mangoes, avocados, grapes, all these fossils. But they're completely gone. This place looks like a desert today. And that's because of the rise of the Andes. They completely eliminated complete ecosystems from this part of South America, which sometimes we don't acknowledge or we don't know about. And this is a fruit and seed that we described early this year. And this one is a seed that today is only dispersed by ants, and is being called macrofossils. And in this case, they have a very lipid-rich type of tissue on the tip of the seed that ants are very specifically attracted. And it is preserved right here. We never thought we'll see that in the fossil record, but fossils always surprise us, showing this ant plant interaction. Now, let me take you to the Panama Canal. And here it is about 20 million years old. There are only sometimes opportunities once in life. And as students, I highly encourage you, you see it, take it. And this is a place that it has this is an event that has happened only twice during the expansion of the Panama Canal. Once was more than 100 years ago when they decided to open the canal. And one was in 2006 when I was a graduate student. And I spent a lot of time in Panama here during the expansion, because to get fresh rock is the most difficult thing when you work in the tropics. But I was there and I was very active, and like I was telling the students at lunch, if you want to find fossils, just break any rock you can until you find something.
That was when the canal was widened? That was when the
Yes.
Panama Canal was widened?
The second time, yeah.
Uh-huh.
And it's very interesting time because South America was still isolated from the northern part. There was still a narrow gap, the seaway, and the plants are going to tell us if they were crossing or not. And plants very early on when I started exploring this area here, and beautifully preserved. And as always, there are things that you don't know what they are. And one of the most common ones were these mysterious black structures that I had no idea what they looked like. Until I cut them and I look inside, and they're full of spores. So it's actually a fungus, a fruiting body of the fungus that feed only on the cane wood that you see today in this Silariaceae group and many others. So here we have one of the only fossil, actually, the fruiting body, actually. And not only that, but flowers, more fruits, grapes, juglandaceae or walnuts, coconuts, an incredible history of dispersion between South and now Proto-Central America. This is what Panama had looked like about 20 million years ago. Some of the plants are still there based on the fossils, but all these animals are completely extinct, and they all came from North America. And the last place that I want to talk about is one that I visited last month. This is a slightly younger, about 20 million years old, 50 million years old. Now, when we think about the Amazon rainforest, we have to think about the Peva system. And this is a period of time where the Amazon did not look like you will see it today. In fact, it was so strange that the ocean, the Atlantic, invaded the inner part of the Amazonia. Imagine having salty water in the Amazon today. Makes no sense, but it happened not only once, but multiple times.
Oh my God.
And during this time, we know a lot about the pollen, but we never found the macrofossils. Except for a few pieces here and there that we described in the last few years. This place is also important for vertebrates. And just this year, it was described a new type of dolphin from the Amazon, from this Pebas formation that has affinities with the Indian freshwater dolphins, the Purosaurus, an iconic type of crocodilian that live in the Amazon that fed most likely on giant sloths that are not anymore in this part of the Amazonia. So I went with my student, Peruvian student, David, and we spent 10 days along the Amazon River on a boat, going locality by locality, collecting rocks. And it truly was incredible. We started in Iquitos and went north and then west, east, excuse me, and fossils everywhere. Lots of wood, qualified wood, roots, leaves, fruit, seeds. Incredible, amazing experience. And we collected almost 300 kilograms of rock. Those are not turkeys for Thanksgiving. These are actually the rocks that we wrap in the best way we could to ship them here to the Field Museum. So my student will have these samples here. So here we're gonna have a chance for the first time to really see the origin of the Amazon, or at least the last 10 or so million years, which today is the most diverse place on Earth. And one of the things that I have to say, one of the sad things when I came back from this field trip was the amount of deforestation that I witnessed in the Amazon River. And not only deforestation, the amount of contamination. There was sometimes literally when we're on a boat, piles of plastic floating on the Amazon River. That was very sad. All right, so that's what I wanted to show you. So a few take-home messages. Think about this Paleozoic forest in Chicago, completely extinct. Very few things that are still surviving. That question that I asked at the beginning, the low latitude region of South America always been a cradle of plant evolution? Absolutely yes, but extinction is a deep force still rooted in our rainforests, and not just because it originated there means that it's going to stay there forever. Completely the opposite, and that has deep implications for what's happening today. Early Cretaceous for us, it really blows my mind. When Paleozoic, I get it, it's too old, it has to be different. But Cretaceous is not that old, but we had no analog. I don't understand how you can have ginkgos, cycas, nitalis, and gymnosperm all coexisting in the forest. Paleoclimate, geologic event, and accidents like the extinction of the dinosaurs have ultimately shaped the evolution of this rainforest. But there are still so many things like plant and animal interactions. And again, think about what if I remove an insect from this forest? The cascade of events that it's going to produce and all the ecological and trophic interactions. And you definitely need more fossils. That's one answer, one message I can tell you. And with that, thank you so much. Before you leave, I want to mention that please, if you want to come and visit us at the Field Museum, we have lots of opportunities. We have internships that are women for women in science internships every summer. But we also have science visiting scholarships for everyone to visit as well. So all that you need to do is contact someone, send us an email, and we pay for all the internships. So you don't have to pay for all the costs, because we know it's difficult sometimes for grad and undergraduate students. And with that, thank you so much now.